Dear students, welcome back to our course Applied Environmental Microbiology. Today we will continue our conversation on bioinformatics. So previously we talked about what bioinformatics is, why we need it, and we revised what we have already studied on metagenomics and why metagenomics is important for us as environmental scientists, students and engineers, and why we cannot make sense out of metagenomics, why it will not help us unless we have strong mathematical and computational tools to make sense out of our data. Today we will continue this conversation further, try to understand about different kinds of options we have in bioinformatics to make sense out of our data. So let us get started. Alrighty, so one thing that we mentioned in previous lecture was that the price of sequencing has dropped steadily following Moore's law, in fact even faster than Moore's law in last decade definitely, and before that, and every now and then, every few years we are noticing we are getting a new generation of sequencing techniques. For example, after the 4th generation sequencing technique which uses pH instead of optical measurement, the next generation technique are actually directly measuring directly measuring the change created by cleavage of a particular DNA, particular nucleotide by passing it through pores. So nanopore would be the next generation sequencing technique. And not only has the price reduced, not only have the techniques improved, but obviously the amount of data we generate has increased very fast. So this is a very important panel here on the right top corner of the slide, that the amount of nucleotide data in billions that we have created in past decades is enormous, and it just continues to rise. So it is safe enough to say that 21st century is a century of biotechnology. And when we say word biotechnology, we are not referring merely to the Department of Biotechnology, the subjects there, basically the technology that is related to life. Now conventionally biotechnology means we are using techniques to change life, to affect life in a way that it suits our purpose. For example, I might modify microorganisms so that they make insulin, and now I do not have to sacrifice thousands of sheep to make insulin dose. And similarly, same is true with vitamins and many other proteins and amino acids that we require for medical purposes and for other civil purposes. So generally this is what we refer to as when we are talking of biotechnology. In this particular slide, we are also talking about all the tools that are either used on living beings, biological beings, or use biological agents as tools. For example, biosensors, which we did in previous lecture, all of them can be collectively called as biotechnology. Alright, so then now let us revisit some of the words that we have, phrases and terms that we used earlier. Genomics. So remember genomics? So whenever you have omics, you means all. So whenever we can sequence all the genes in your sample, we call it genomics. When we are referring to all genes and their analyses, it's genomics. And now here's the thing, genomics does not only mean genes, because there are non-genetic areas too, the non-genetic regions, non-coding regions in the chromosome, in plasmids, so including all of that. Now here's the thing, new sequence information is being produced at an increasing rate. The contents of Gen Bank double every year. This is very fast and crazy fast growth. Next is microarray. Even in biosensors, I briefly covered microarray, but microarray is not used a lot now. It is out of the picture. Now it has been replaced by RNA-seq. RNA-seq will help me sequence RNA directly, and I don't need to have a microarray to await the hybridization. That's a good point to mention here, that microarray was typically used for rapid detection of RNA. Now if you remember the central dogma, you will remember RNA is a better representative of what's really happening right now, whereas DNA also includes the potential for future and the baggage of history. Now in proteomics, that is analysis of proteins, all proteins, global protein analysis generated by large mass spectral libraries. Metabolomics, we are looking at metabolites. Global metabolite analysis, 25,000 secondary metabolites have been characterized. That's an immensely large number. So I hope this gives you an idea of how much data we are generating every year. Now Now doubles every year, you have to understand it grows really fast, the genomic information. Most of the environmental scientists, they nowadays focus on genomics, proteomics, and metabolomics. Mostly genomics and proteomics. So metagenomics, I mentioned this in previous lecture, so let's revisit again. It tells me who is present and what they are doing. So because I have the genetic sequence, I can generate assemblies. Let's say this region is your 16S rRNA gene region, and this is the sequence that you have created, this is the sequence that you have created. So we can align it to our database, and then we can get information of who is present. There are other ways also of finding of taxonomic identity of the sequences and the assembled scaffolds, or assembled contigs, or assembled genomes, but this is the most popular one. And we can also look at the other genes that are present. For example, if I am interested in antimicrobial resistance, I might say align this with a database called CARD, and Comprehensive Antibiotic Resistance Database. And then I will know what kind of antibiotic resistant genes are present. If I am interested in cellulose degradation, I might look at KZ. Carbohydrate, this KZ database involves all enzymes, all the genes that are involved with carbohydrate degradation. And then I might get genes, okay, what genes are present here that are associated with carbohydrate degradation. So not only do I know who, but I also get information of their function. Basically, what are they doing? Alrighty. Now the beauty of metagenomics, as we mentioned earlier, is that it is cultivation independent approach. So we do not need to cultivate it. We don't wait, because remember from last lecture, 99% of microbial communities, we suspect members of microbial communities are non-cultivable even if they are viable. So they will survive in the environment, but we can't grow them in the lab, despite so many advances in microbiological tools. So metagenomics helps us there. For example, we can take samples from very different, very diverse microbial communities, and we can extract their genomic DNA. DNA extraction is a very simple process nowadays, because we have a kit for most kind of samples. Like you have solid samples, you have soil samples, you have fecal matter, you have blood samples, you have a tissue sample, you have bacterial sample. You can take all kind of samples, and you will have a kit for it, and then you can extract DNA. And after you have extracted DNA, what we do is we get heterologous genomic DNA, and then this can be broken. Well, this is the cloning part, by the way. So what we can do is we have restriction digested vector. Now this restriction digested vector will make sure that these DNA fragments now will be transformed into the competent E. coli cells. Now one thing you want to know, because here we are doing cloning, typically there is another step here, which is from the we don't like to usually we don't like to clone the entire sequence. What we will do is we will amplify a region of it. So we will use PCR, and the kind of cloning kit that I am familiar with, we leave a sticky end with an A overhang. So basically it is like at the end, if you have here T, you have A, you have G, C, T, A, T. So at the end we will leave an A overhang. There is nothing here to match with A, nothing. So this A end is a sticky end, and this will attach with your vector. So your vector will attach here, and now your vector is attached to it. Now this can easily be cloned into competent cells. So you have cloned it into competent cells. Now your competent cells are expressing the protein you are interested. The protein might be secreted, so you can extract this secretion. For example, the protein could be some of medicinal values, then you want to do it at high quantities. The other possibility is that when you have done the cloning, what you can do is, now remember, the red one is your vector, and the blue one is here the sequence that you wanted to clone. Now each of your successfully transformed E. coli cell will carry the same thing here, like this. Now let's say you want to sequence this. Now in order to sequence this, what you can do is you can amplify regions of this particular plasmid, and you can sequence them. Now here you will get very high fidelity, in sense that you will get very good results for your sequencing. If this is useful for typical Sanger based sequencing technique, in metagenomics we skip all these steps. We don't need to do this. Remember, cloning, you take nearly 1 day to set up your cloning, and then you have to leave them overnight, and then you send them for sequencing, and then you get your data, and then you look at your data using your tools. It takes at least 3, 4, 5 days, depending on what kind of resources are available to you and whether anyway. But in metagenomics we skip all these steps, and we go from genomic DNA extraction directly to quality check, quality control, and then you can send it to your sequencing agency, and then they will do the next steps for you, which don't look like this. Alrighty. Now the trouble of today's world is that we have so many omics around us. We have metabolomics that's giving us information on what metabolites are present, proteomics that's telling us about proteins, next generation sequencing are giving a lot of metagenomic data, microarray not popularly used now anymore, but even microarray kind of techniques give you a lot of data, epigenomics, we have talked about them. They give you data, clinical trial data now you have, and then you need to also visualize your data. So all this data is very hard to visualize. As a result, we come in fix. So how do we handle this large amount of data? That is a very pertinent question. And this is a very nice cartoon. Here you have a puzzle that you need to fix with 3 billion pieces. So you have seen the jigsaw puzzle where you have to fix them, and it's really impossible to even understand where to get started from. And here's a joke, one of the scientists is saying, I think I found a corner piece. The answer is bioinformatics and internet. So if you can use bioinformatics, then the computer will decide which pieces go well together. Remember what I told you about sequencing and assembly in the previous lecture? You create millions of these sequences. So these are your millions of sequences, so 3 billion pieces. So you have created 3 billion sequences. You don't know how they fit together. How they fit together, how can they be stitched together to create greater length of sequences. Don't have to worry about finding the corner piece. What you can do is you can use bioinformatics tools, internet based tools, and what they will do is they will stitch it for you, and you, if your sequencing data is good enough, you might even get the whole genome sequence. Now how does this work? Let's say you have just cloned a gene using the typical way. Then what you can do is you can check out if it is already present in the database, then what is its accession number, what is it annotated. After that you can look, are there similar sequences in the environment that other people have reported, what is the percent identity, what is the family member. And in one of the lectures on bioinformatics we will be going through NCBI, and I will show you how to find out percent identity and find out about family members. So here's the thing, if a sequence that I have cloned is very similar to another sequence that is already known, so I know the percent identity, then percent similarity with a known sequence, then I can get an idea of what kind of microbe, what kind of gene this is. So this is very important. And the next step is I want to find out, are there conserved regions in it. If you remember from 16S rRNA analyses, one of the beauty of 16S rRNA is that it has conserved zones. So let's say this is the hypervariable region or just the variable region. So what we can do is we can align all 16S here, because the conserved region should be same in most of them or all of them, very similar. So this is very similar. So they serve us for aligning sequences with each other and with database, and then we can see, okay, this one belongs to Clostridia, okay, this one belongs to negativity cuties, this one belongs to something else. So this kind of differences that we can then analyze, but we need to have something to staple them together and say, okay, this is my landmark from which I am comparing. So that's why alignment is important, domain analysis is important, and post that you want to understand evolutionary relationship. Already now you know different kinds of bacteria were detected in your sample. What is the evolutionary relationship? How close are they in relation to each other? So to do that kind of analysis, you can make trees. So for that you can do cluster dendrograms. It is called, the one that is used most commonly is called hierarchical clustering. I encourage you to install a software called R. It's a statistical software, and I encourage you to download a package and install it called pvclust in this software. So once you get the R software, all you need to do is you need to download the package pvclust, and once you have this package, you can very easily do hierarchical clustering of your samples, and they will be aligned together in basis of their similarity. Now if you are interested in phylogenetic tree, then you have other options with you, like for example you can use ribosomal database project. This is an online, so if you can just Google RDB, you will get information on how to create phylogenetic tree. So once you have phylogenetic tree, it will look like this typically. It will have dendrograms, and then whenever you see different microbes coming from similar stem, we can say this is the common ancestor of all these. And then if this is the initial stem, then this is the common ancestor of all these. So this is evolutionary relationship. And then we have protein characterization. So what you can do is you have the gene, let's say you also allow the competent cloned, in which you have cloned that competent bacteria to express the protein. You can actually analyze the protein, or if you have not expressed the protein with your genetic sequence, you can do modeling to find out what will be the conformation of this protein, what will be its putative activities, and you can get a very good information on protein characteristics. Next is you can also get other kind of information. You can find out expression profile. You can also look at mutants. For example, there was a study reported in 2013 I think, 2014 or 2013 in Nature, where what they did was they fed monkeys that had tuberculosis anti-tuberculosis drugs, and some of the monkeys had tuberculosis, I think some of them did not, and then they noticed over time how did the tuberculosis, actually all of them had tuberculosis, yeah, how did the mycobacteria that causes tuberculosis to these monkeys, how did that change over time. So this kind of analysis can also be done using metagenomics or using genomic sequencing techniques. So basically you will have at time t equal to 0, you will have a particular sequence of mycobacteria. At time t equal to 10 days, you will notice that there are some changes here, some single nucleotide polymorphism, some shifts or deletes or additions, and then time t equal to 100 days, you will notice more shifts, more changes, and then this way you can get an idea of the what is the evolutionary relationship. You can also get an idea of how are the sequences changing with time. But so all these tools are available for us and online. You can look them up if you know the term what you're trying to do. If you're clear about it, you can look them up and solve. But remember that a critical failure for current bioinformatic tools is that we do not have a single software package that can do all of this. So there is no standalone single software package that you can install in a computer and do all these analyses. So this is something that there is room for improvement. So if you're interested in coding, if you're interested in computers, I highly encourage you to check out what's happening in bioinformatics, what are the limitations currently, and how we are trying to improve them, because there is a lot of scope of research, of creativity, and even entrepreneurship in bioinformatics. Some of the tools that are very popularly used for DNA databases that are popularly used by scientists, researchers, students across the world to compare the data with, to align it, to annotate it, are GenBank's NCBI, then DNA database of Japan, DDBG, then TIGR, EAST for East Genome, and then we have a microbes, and then there are others that I would like to say. There is the Green Genes, and then there is Silver. I encourage you to go through them too. The advantage with Green Genes is that it's highly curated. What it means, the highly curated word is that the quality of the sequences is checked over and over again. So if you get a good match, you can be very sure that it is that match, and there is very less chances of error. For specialized databases, we have ESTs, which is for express sequence tags, and many more for antimicrobial resistance. We have CARD, as I mentioned. For cellulose degradation, we have KZ, and so forth. All you need to do is Google and you'll find out the database that will fit your requirement. For protein analysis also, we have a lot of databases. We have SwissProt, which happens to have very high level of annotation. We have PIR, Protein Identification Resource by UniProt. It is the most comprehensive database in the world on proteins. Then we have Tremble, which means Emble, by the way, and it's like the pre-version of SwissProt. So basically the sequences that have not been included in SwissProt yet are present here. Then again we have NCBI. Now this is very interesting what NCBI does. So if you look here, NCBI is on top when it comes to DNA databases. So it has one very nice technique where it will translate your nucleotide, your genes, genetic data into proteins. So if you know where the open reading frame is, you know where the start codon is, you can start translating it very easily by just looking into the table. So what you have here is ATGC, you will have here as your proteins now. So the protein version of your GenBank is called as GenPept, and then we have Protein Data Bank, again very important. Alrighty, so dear students, in summary, your metagenomics will help you take your sequencing data, you run it through the computer, so that it does not break your computer but makes sense to you, and you can get meaningful data to answer environmental challenges, to answer the questions that you have been asking. So that's all for today. In the next lecture, we will talk more about bioinformatics. Thank you.